Let's talk about some specifics of epithelial tissue, so the different classifications. First, what I want to do is just list them, and then I'll talk about which ones I care about the most. So first, we're going to go through the different types. Basically, if you remember your simple and your stratified, and your 3 cell types, squamous, cuboidal, and columnar, you put those different combinations together, you're going to have most of them. There's 2 additional that don't fall into one of those categories. One you saw already, pseudostratified. Okay, so let's go through these. Simple squamous, so 1 layer of really short cells. We'll see example pictures of this in a moment. This is going to be where you want transport, really thin membranes. The membranes aren't thinner. It's going to create thin membranes though. So for example, the membranes around your internal organs. Also the alveoli of the lungs and the capillaries, the linings of the capillaries, because you want oxygen and carbon dioxide to be able to pass across those cells easily and quickly. Stratified squamous, so layers of short cells. This is going to be good for protection. So esophagus, ureter, skin. Simple cuboidal. The example of this we'll see is the kidney tubules. We're going to have absorption and secretion passage across these cells, but the cuboidal is a little bit thicker than the simple squamous. Some of the kidney tubules are simple squamous. Stratified cuboidal, this is an example of a rare tissue type. Many glands are stratified cuboidal. I don't have this one in bold. I won't talk as much about glands in this video. Simple columnar, this example is the intestines. So a single layer of tall cells where absorption is going to occur across those cells. Stratified columnars, also those glands. So stratified cuboidal and stratified columnar are less common and typically make up glands. Transitional is one of those that does not fall into the category, those basic categories. This is the bladder. It's stretchy. I'm actually not going to talk about it a lot. It looks a lot like stratified squamous, but it stretches. The bladder needs to stretch. Pretty useful thing. Lastly, we've got pseudostratified columnar. This is the respiratory passageway, trachea, for example. This is going to be that ciliated stuff that looks like it's stratified, but it's actually pseudo-stratified, so actually a single layer. Okay, so let's talk about each of these with pictures and comparing them. So first of all, I mentioned this already, but why are the alveoli of the lung and the small blood vessels, for example, capillaries, also the small venules and arterioles, do those have simple squamous epithelial tissue? That looks like this. So this is a single, it's kind of hard to see where the separate cells are, but there's separate cells here, around here, very, very thin cells. So this would be the basement membrane right here. This is the apical surface right here. Basement membrane, apical surface. Sometimes you can't see the connective tissue very much, especially with the alveoli. So why? Why is this so thin? There's still 2 membranes here, but the cells themselves are thin. This allows for quicker diffusion. Remember diffusion distance. The distance that something has to diffuse across influences diffusion rate. We want oxygen and carbon dioxide to be able to diffuse across both the alveoli and these capillaries, this is a venule in this case, very quickly, as quickly as possible, because that's what respiration is. And so a thin lining is helpful. There's actually a kind of picture of that here. This is a schematic of those same epithelial cells that are part of the alveoli and the capillaries here. These thin, 1 layer of thin cells allow oxygen and carbon dioxide to pass across quickly. The other big example of the simple squamous epithelium is in some of the linings around the organs, those membranes. And we'll come back to those membranes next week when we start talking about skin. I'll review them briefly. On the other hand, why are the skin and esophagus composed of stratified squamous epithelial tissue? I think you know already, is for protection. So the skin itself is an organ. We're talking about skin next week, but the top layer is made of epithelial cells, stratified squamous. So that's this layer right up here. So the top layer of the skin, the epidermis, which is often what we think of as the skin, is all stratified squamous. It's actually keratinized. What that means is the cells produce keratin, a protein, and that keratin, those keratinized cells, is basically what's on top. They're super protective, they're waterproof. Talk more about skin next week. But that's what makes the keratinized skin cells different than the esophagus. So esophagus, ureter, vaginal canal have stratified squamous as well. They are not keratinized. They don't have that same waterproof layering. Either way, this is for protection. Very protective layers of cells that can slough off, right? If you get injured, your skin heals quickly. Same with your mouth. And dead cells are constantly falling off of your skin. So, layers for protection. Okay, what about the kidney tubule? Why is this composed of simple epithelial cells? Simple cuboidal in this case, but I kind of want to focus on here to make sense of it. So I'm trying to connect structure to function to help you remember these tissue types that are here. The kidney tubules, so you take the section of the kidney, these little tubes are called nephrons, are all in your kidneys. This is where your blood becomes filtrate and then becomes urine to go out of the body. You take a section of these, besides this part down here, it's all, this down here is actually simple squamous. Up here, it is simple cuboidal. And this is, so here would be a long, like a schematic of it in a longitudinal section, like this one here. And here is our cross section. So it's cutting those tubes at a cross section. And here we want to be able to have absorption and secretion occur. We want to have a thin layer of cells, one single layer, so that the urine, the parts of the urine we want to keep go into the body, and we also can secrete extra ammonia and wastes out into the urine. So basically transport, talking about here, we need to have transport. So what we've done so far here is explain why the epithelial cells would be stratified versus simple by comparing their functions, and predict whether a body part would contain simple or stratified squamous epithelium. So, and I want you to be able to predict, like, okay, this is the function of this body part, what's it gonna have there, why? So here are the three specific examples that I talked about: the alveoli and the small blood vessels, the esophagus and skin, and the kidney tubules. Okay, second part of this is the pseudostratified columnar epithelium that I want you to know. So this is located in the respiratory passageways, trachea, as well as some other ones as well. This pseudostratified is unique to the respiratory passageways. It's pseudostratified, which means it looks like it's stratified, but it's actually a single layer. And pseudostratified columnar is always ciliated, and it always has these goblet cells. This relates to its function. So respiratory passageway, what is the trachea for? Well, first of all, it's for getting air in and out. It's a tube, literally just a tube for air to go through. But it also is designed to get stuff out. You don't want junk, debris, bacteria, mucus going to your lungs. So the trachea produces mucus, and these little cilia are cytoskeletal extensions that can actually move, and they propulse the mucus and the debris out of the respiratory passageways. We won't be talking about these non-ciliated types, so we're going to only talk about the pseudostratified, ciliated pseudostratified columnar epithelium. This would be what on a section of the trachea, what that looks like, the lining that is pseudostratified columnar. So single layer. Down here, these are other tissue types. Hyaline cartilage we'll come back to later. So zoom into that. Here's our basement membrane, single layer of epithelial cells. In lab, make sure you know what zoom you're looking at. Try to think about, where are you? Are you here? Are you looking at just the epithelial cells? And then this is a nice one to contrast to simple columnar. They look similar under the microscope. The difference is because the simple columnar also looks like a single layer, just like pseudostratified columnar do. However, so I wouldn't actually go by cells when you look at these under a microscope. I would go by the shape of the entire organ. So looking at by eye, is this trachea or is this intestine, which you'll learn. And then also these microvilli versus the cilia. The microvilli are also cytoskeletal extensions. They do not move, and they are a lot smaller. So you cannot actually see individual microvilli. That's why it's also called the brush border, because it kind of just looks like a brush on the top. So it's going to look a lot different than your cilia, which you actually can see individual ones just barely. So in this case, these microvilli are designed to increase surface area for absorption. So the intestines designed for absorption. These cells are good at absorbing because they've got a large surface area. And then down here would be the basement membrane. The nutrients would go into the bloodstream from here. So what we've done here is compare and contrast the epithelial lining of the intestines and the trachea as our two examples of pseudostratified columnar versus, simple columnar versus pseudostratified columnar, and the importance of the key structures. So some of those key structures being microvilli, goblet cells, and mucus, those two in the trachea. Some of the functions, cilia, also in the pseudostratified columnar. All right, that's it for epithelial tissues in lecture. You'll see them in lab. And next is connective tissues.